Thank you. Thank you so much. Good afternoon. It's very exciting for me to be here. It's my first JGI meeting that I get to attend, so really cool technologies and area of focus. Actually, today I get to fill in for the head of discovery at Indigo, Barry Goldman. He couldn't be here, but that means that it's a great opportunity for me to speak about really a couple of projects. So the title of my talk does a good job in describing one of the many projects that I get to work on at Indigo as part of discovery and R&D. But before we dive down to this project, I would like to take a few minutes to introduce Indigo to you, to give a little bit of a background and put things in context. So Indigo was founded 4 years ago in 2014, and with a vision. And this vision pretty much stemmed out of the human microbiome field, that microorganisms associated with plants could make a big impact on plant health and overall fitness. And such microbial products could be ultimately used to really improve on the farm profitability. So we really put the farmers first. Our headquarters is in Boston, precisely in Charlestown, from which I flew in a couple of days ago, running away from the snow. We have also a big presence in Memphis, where most of our commercial and agronomist folks are located. We recently opened a facility in R&D in North Carolina to do more research and development over there. And we really think about improving farm profitability anywhere in the planet. So we really think globally. And therefore, we have also international offices, which are in Australia, Argentina, where Barry is now, and Brazil. And these past few years have been tremendously exciting for us. I personally joined a couple of years ago, and I must have been employee number 35. Now we are 300 people. And so it's been really, really a journey, very fast-paced and exciting. Along this time, we also managed to put some products and launch products for cotton, wheat, corn, rice, and soybean. So at Indigo, we have quite an ambitious mission, which is harnessing nature to help farmers sustainably feed the planet. So starting with the farmers, and we heard a very good talk yesterday, by the way, about agriculture, modern practices, and use of the land. So if you think about growing food for ever-growing population, really, it's a complex process, multiple steps. There are many challenges, such as limitation of clean water, limitation of, and degradation of arable land, and also climate change. So farmers are really, they really have a tough time sometimes in taking all those decisions in terms of what to plant, when to fertilize, when to apply pesticides. And then they have to deal with a lot of unpredictable factors just from the weather, for example. So what we strive to do is really to use the microbiome to its full potential to really try to improve on the yield, and so in the farmer, really make a difference in the farmer economics. Now, shifting to the central part of the slide, another core value that we really embrace is sustainability. So we would like to be able to produce enough calories and food for everybody, increase the yield, but doing that in a sustainable way. So we think the microbiome, again, could be of use in terms of replacing the bulk of pesticides, insecticides, and fertilizers that have been used as part of practices in modern agriculture. And ultimately, if we manage to help the farmers improve their economics, and doing that in a sustainable way, the impact could be really terrific. So all the consumers, which is us ultimately, could have access to food which is produced in a more sustainable way and healthier food. So at Indigo, we focus on the microbiome and think about plants. Plants have co-evolved with the microorganisms for hundreds of millions of years. And they really are co-evolved, and they are in an intimate relationship where the plant offers a house for the microbe, and the microbe can take advantage of nutrients, water, and then the microbe itself can benefit the plants. But again, as part of the as a result of modern practices in agriculture, the microbes have been depleted, so we find much less presence in the soil, and also the cultivars themselves, what gets planted in our fields, is really far away from their ancient ancestors. And so the microbes have been depleted, and what we are trying to do at Indigo is really, most what we do is to try to put the microbes back where nature intended them to be, probably all along. And we can argue that many plant fitness traits can be influenced by the microbiome. So we can refer to both biotic stresses and abiotic stresses. So think about plants that live, for example, in the desert or in salt water, or experience really high temperatures or really cold temperature. And those plants, we find examples of plants that not only survive, but they really thrive in those environments. But it's not that they do that all by themselves. Actually, it's quite the opposite. So microorganisms, endophytes that live within these plants can indeed provide a benefit and help out in, for example, tolerating drought, heat, cold, or infestation by pathogens such as fungi or bacteria. So in order to help farmers, we built a pipeline. And this pipeline comprises quite a few steps. First of all, we acquired thousands of strains in a few ways. So we have in-house effort where we source strains from the fields ourselves, but also we work closely with collaborators, and we get strains from them as well. We also, when we acquire strains, we collect a bunch of metadata, which is powerful in making sense of all these strains and their features. We do a lot of sequencing. We sequence all the strains that we have in the collection in some sort of way. So sequencing is really a cornerstone of our pipeline. And then we start testing, screening. We have a few ways of doing that. So we first generate hypotheses. We select the strains out of our collection, and we run high-throughput assays, and we test them in the greenhouse. We test them in the field. And we really make the farmers our partners, and we get them involved with the testing as well, so that we really are transparent in our approach, and we share both the risks and the benefits associated with this testing. And we go through many iterations of this pipeline so that every time that we go back and we select new strains for testing, we gain more information from the previous year. So this is really like Indigo in a nutshell. And what I would like to do before switching to the project that I'm going to talk about sequencing 200 fungal strains, I just want to make sure that I make it very clear that the project I'm going to talk about is not about making products out of those strains. It's really an academic project in its nature. We are interested in understanding more about the microbiome and contributing that to the community. So we are really much interested in making all the data available and publishable. So what I'm going to talk to you about is accelerating insights into genomic drivers of fungal symbiosis with plants. The biological question that we had in mind when we started this project was about what makes an endophyte, so in this case a fungus which lives inside the plant, beneficial. And what are the differences, the fundamental differences between closely related species or genomes of fungi, where one of the two in the pair is a pathogen and the other one is an endophyte that can have really beneficial effects on the plants. So you can see a couple of examples in this slide of genera that really have been known to be very famous or infamous pathogens like Fusarium and Coletotricum. And people have been focusing on describing these pathogens for a long time. There is a lot of information on those, and rightfully so. But at the same time, there are strains and species within the same genera which are not at all pathogenic. They are endophytes, they live in the plants, the plants might not manifest any symptom. Or on the other end, like they can be friends and not enemies of the plants, so they can provide, overall better plant health. They can help against stresses of biotic or abiotic nature. So we really wanted to see what makes a pathogen, what makes an endophyte from the point of view of comparing these closely related species and look at their genomes. In order to address this question, at Indigo, we piloted a study, which is very collaborative in nature, and we got a couple of academic collaborators involved, and our sequencing partner is Illumina. So we basically leverage all the expertises present either at Indigo or in academic lab, and we divide and conquer. So we acquire strains, we grew them, we figure out the best way to do so, keep them happy, did DNA extractions, library prep, sequencing, and ultimately we are now focusing, it's early days, we just got started with analysis and comparative genomics. So our partner are Illumina for the sequencing, and at UC Riverside, the Stagis lab, and Arizona, the Arnold and the Euren lab. So in order to sequence this genome, we wanted to carefully select them, and we targeted sequencing of closely related beneficial as well pathogenic fungi. And we obviously focus on species which are agriculturally important. And we did a very phylogenetically controlled selection where we try to follow as much as possible principle of microbial ecology, evolution, and comparative biology. We focus on focal classes which contain the most beneficial as well as the most devastating fungi for agriculture. So we leverage strains present in collections from all over the world, really, the US, Europe, Asia, Africa, to really have a wide range in terms of sourcing. And we pick both Dotiomyces and Sordariomyces. And this is really with the aim, once again, I want to stress on the fact that we want to publish this data and make it available for the community. So there was a lot of work that went into just getting all the strains, growing them up, figure out the best way of extracting the DNA. I'm not going to talk about that. Once we got the reads back from Illumina, we just ran our in-house analysis pipeline. And so we did the usual trimming, QC quality. We did de novo assembly with a couple of algorithms, gene prediction, and ultimately functional notation to see, okay, what are the genes predicted to do. Sequencing overall resulted in good quality assemblies. We had a few samples that didn't work out. We might go back and resequence them. But overall, we got good quality assemblies that we could work on. And based on a couple of stats here, the N50 as well as the BASCO, so we were able to retrieve enough genes in terms of numbers, expected numbers in the assemblies. Now I'm going to focus on an even smaller section of the 200 fungal genomes, because again, it's early days. We just gotten started with analysis and taking a closer look. In particularly, here is a section that comprises, again, fungus, which is known to be a pathogen, Bipolaris and Coccobolus. And it's a pathogen that causes quite important diseases in the field, especially in the family of the Poaceae, like Bipolaris bidis, so rice, wheat, maize, cause leaf spots, rots. And so for this particular genus, we selected species of pathogens, but also species that are not known to are endophytes, and they don't cause any harm. In the reads we obtain from sequencing built into similar size assemblies. So here I plotted the two members of the pairs, endophyte and pathogen, in green and red respectively. And I compare the assembly size in megabasis and the GC context, and overall the size of the assemblies matches, and also the GC content, just to get a first understanding of the genome stats that we were getting. And then we also built a tree based on 500 single copy shared genes that were randomly selected from the thousands genes from the BASCO analysis to really compare how phylogeny was aligning with the evolutionary relationships, which was deducted by comparing these genes. And you can see that each pair, within each pair, endophyte and pathogen cluster really well together. We also look at the gene content, obviously. So here you can see the orthologous genes content is mostly conserved. So we didn't anticipate, and we are not seeing so far from this small part of the analysis major differences. So there are a lot of genes which are expected, and they are indeed conserved in all the samples and shared also among subset of samples. But indeed, there are unique genes belonging to each strain, and we think those are the places where we really want to dig out more, and we are curious to see what we'll find. We also look at endophyte and pathogen pairs in terms of protein content. And we basically, with the MDS, we ask, okay, how do these strains compare to each other? Which one is more similar to what? And indeed, you can see again, here the triangle represents the pathogens and the circle is the endophyte. How each pair, within each pair, the endophyte and the pathogen look similar to each other more than to outsider, another pair, with a couple of exceptions. So the ones in green and the ones in pale blue. And there we don't know the reason. We can speculate that perhaps those two are really indeed quite different from each other, or perhaps they are not so close as we thought in terms of similarity between two species, and we haven't really sampled much in that case. We also looked, and this is the final piece of results that I have for you today. We look at the CAZymes, sorry, actually, I skipped one slide. We look at the polyketides. So polyketides are involved in secondary metabolism. So we look at, they are encoded in the genome, but these multi-domain enzymes, which are polyketide synthase, and the acyltransferase domain is one of the many domains. So here we wanted to see what's the presence of these domains using PFAM. And what we observe consistently, if you look at the pairs, is that for each and every pair in the pathogen, we found more abundance, so an increased abundance of these polyketides. And this is, again, probably, it's early days, but we can speculate that this secondary metabolism really play a role in the evolution of these strains, and they provide really, you know, an arsenal of bioactive products for these pathogens to use with the plants. And now, last but not least, the CAZymes. So we also look at the carbohydrate metabolisms. So in this case, this map includes only the families in which at least one species has had 10 or more members. And we have examples from the glycoside hydrolase as well as the carbohydrate esterase, and also the auxiliary activities, which are redox enzymes, which act with the CAZymes. This heat map is displayed quite a similar pattern, yet there are some distinctions that we can make. Again, it's early days, so we have to investigate more. I think either way, it would be interesting to see what we find, and it may be different based on the type of pathogen and endophyte that we are looking at and focusing on. So in conclusion, I told you about Indigo, and I told you about us doing research and sequencing 200 fungal genomes. And we just started taking a peek at their genomic content and features. We look at their overall protein content. We look at secondary metabolites and CAZymes. And we notice both similarity, but yet distinct patterns. And there is really a lot more to come because we want to include all the genomes that we sequence, of course, and look more into details. And we want to also contrast recent genome evolution among pairs of endophyte and pathogen, as well as doing a global analysis to identify trends in the genome evolution. We haven't looked at transposable elements, for example. We haven't looked at other gene clusters. We have not looked at effectors, especially in the pathogens. So a lot more to come, and I hope I'll be able to share soon. Finally, I want to acknowledge all the people that have been working on this project and are still working on this project. From the West Coast, Jason at UC Riverside, who's providing guidance in the way we analyze the genomes. Jason and Ryan at Illumina for the sequencing. At University of Arizona, a couple of labs, the Arnold Lab, Betsy and Kayla. The Euran Lab, Jana is here in the audience as well. And at Indigo, my colleagues Swati, Luis, Nicole, and Barry. This is a very fun project. We also got to take fun trips to San Diego at the Illumina site. Kayla and I went last year, and also Swati went to Betsy Lab to help out with extractions. So really a fun collaborative project. And I thank to all of you for your attention.